Proteomics is not only identification of proteins, it's also analyzing the function of proteins. That means studying protein-protein interactions, studying protein modifications, and that's more my part in the platform, because my lab has an expertise in expressing proteins in bacteria and in other organisms, and then studying by diverse method interactions between proteins.
By measuring the mass, we can understand how these molecules are regulated and how this regulation changes in a different biological setting or in a different clinical setting or in a different medical setting. But it's not just the scale, it's also basically allows us to take these molecules apart, and by taking them apart we can better understand how they are built up. And with this kind of machines we can study all the proteins of a cell type and can understand how this is regulated in a disease setting, for example.
It's very, very diverse. We have projects where we analyze the brain lipids of Drosophila, a small insect that is a model here in the Department of Biology. We analyze the vitamins, the lipophilic vitamins like vitamin E in my own research group, in plants and in vegetable oils. We can analyze some lipids that are important in the symbiosis between fungi and plants. And these lipids are essential for the symbiosis. So it's very diverse. We also ran previously some samples of vineyards treated in the natural way, or the classical way I should say, with a lot of chemicals, and vineyards that are not treated with chemicals. And we analyzed the soil to see the diversity of the microflora. And we observe very large differences. So basically lipids are building blocks of cells, so we can analyze huge varieties of samples.
So we really have a state-of-the-art expertise and technology from which all customers can benefit.
They basically do not have to buy a machine. They can come to us and we design with them experiments and we help them in interpreting the data that these mass spectrometers generate.
The instruments and also the analyzers rely on a high mass accuracy, so that what I have to do is do always a mass calibration of the instruments so that they produce good data. And of course often the samples are a little bit dirty, so they have to be cleaned regularly.
We help in the experimental design and then help us in sample preparation, perform the data acquisition and data analysis, and then also help in the data evaluation.
If you look at the breakthrough papers which come out in the last 5 years, most of them have, at least in part, some kind of data that was generated by a mass spectrometer. So definitely without this breakthrough science is much more difficult.
You never know what you're going to get, and it's always pushing you into a new world. Of course you have your expertise, and this is really the basis of this platform, but intellectually it brings you in a lot of different worlds, and this is really passionate.